Hello, good morning, friends. Today topic is very interesting. It is to prove that DNA is the genetic material of heredity. It is sure that genetic material DNA is the sign of heredity. It is the cause of heredity. DNA is the genetic material which is transferred from one generation to other generation. As we know today, it was proved by some experiments. The first experiment, which was given in 1928 by Griffith, it is called Griffith experiment. The diagram in front of you shows, I will explain to you, that there is a transformation of one type of bacteria into other type of bacteria, showing that DNA is the genetic material. Now, there is a bacteria for pneumonia that is called Streptococcus pneumoniae. Streptococcus pneumoniae is of two strains, two types, smooth and rough. Smooth causes pneumonia and rough causes no ammonia. What did the experiment? I was telling you about the Griffith experiment. Griffith took the mice and injected first mice with the living cells of smooth bacteria. It was injected with the smooth bacteria, the mice was died. In second stage, the other mice was given the living bacteria of having that very rough bacteria. That was not poisonous. The cat was not killed. In third case, he gave the rat heat-killed virulent bacteria. Virulent means pneumonia causing. That very rat did not die because they were heat killed. In fourth experiment, he mixed the heat killed bacteria and rough bacteria. Rough bacteria and heat killed smooth bacteria. I mean non poisonous rough cells and poisonous S cells, but heat killed were mixed and made that very injection. The injection was given to this very rat, but now the rat died. It proved that the DNA was present in the heat killed S cells, but when it was mixed with the rough cells, non poisonous cells, the non poisonous bacteria changed into poisonous bacteria. And he has made a result that DNA is the genetic material of the transfer of heredity. The heredity dependent upon DNA. DNA is a chemical base of heredity. This was the result of the Griffith experiment. Another experiment is by Hershey and Chase in 1952. He took the bacteriophage, T2 bacteriophage. Bacteriophage has this shape. It is made of head, tail, and tail fibers. And head is made of DNA and protein coat. This protein coat is made of sulfur, and DNA is made of phosphorus. This is tail and tail fibers. This is structure of the T4 phage. What did he do? He do some experiment with the T4 bacteria, T2 bacteriophage. This is called bacteriophage. He made the experiment with E. coli bacteria. He infected the E. coli. This diagram shows that he proved that DNA is the genetic material. He took two cultures and he made in one culture the T2, bacteriophage with radioactive sulfur. It means protein coat was radioactive. In second case, the phage DNA was made radioactive P32. I mean that two set of bacteriophage, two set of virus. One set of virus was given radioactive phosphorus and other set was given radioactive sulfur. It means in one case the protein coat was radioactive, in A case protein coat was radioactive, in B case the DNA was radioactive, because phosphorus is the part of DNA and sulfur is the part of protein coat. Now, the bacteriophage was entered the E. coli and multiplied. Then he took the supernatant and the liquid and made centrifugal experiments of radioactive phages. He took the radioactive phages and made the experiments centrifugation, that is a physical process, means shaking. Centrifugation means shaking. And by shaking, the protein coat was separated with DNA. In what was the result? In one case, at the bottom, the supernatant was the protein coat and the pellet was heavier at the bottom. In A case, the pellet was not radioactive because the protein coat was DNA. In second case, the pellet DNA was radioactive and the base was radioactive. In one case, the DNA was not radioactive. In other case, DNA was radioactive. In one case, the transfer of the DNA was, but that was not radioactive. In second case, DNA was radioactive. In the newly formed bacteriophage, some were in next phase that the two E. coli, in the two E. coli, one E. coli DNA was not radioactive in the daughter bacteriophage. Daughter virus were not radioactive. In other case, the daughter virus was radioactive because phosphorus radioactive was present in DNA, not in DNA, and DNA was transferred from one generation to other generation, not protein coat. Thank you. See you morning. You study this topic. Interesting.